And so today I am going to be talking about visual processing in different animals. And so specifically, we're going to talk a little bit, we're going to talk a lot about fruit flies and then a little bit about humans and a little bit about mice. Let's see, but before we do that, I want to tell you a little bit about myself. So as Lester said, my name is Caitlin and I study physics. And I'm from Florida originally. I have a very wonderful cat named Wednesday, and I love arts and crafts. And I always think it's important to talk about the things we like other than school because we have lots of things, not just school, that we do in our day to day. Although I will say, when it comes to school and academics, absolutely love science. And I love teaching science to lots of kids too. And so I am what's called a graduate student. And what that means is, so start off in school, in elementary school. And then, as we know, we go to middle school and then we go to high school. This is actually a picture of me in my high school physics class. And then one of the things we can do after high school is we can go to college. And at college, we have the opportunity to study something a little bit more specific, right? In elementary school, middle school, high school, we're studying lots of different topics. And then in college is the first time we can start to really focus in on something. So in college, I decided that I wanted to take a lot of classes and learn a lot about physics. And this is actually another picture of me doing an experiment for a physics class. And then after college, lots of people will decide to go to get jobs. Some people might go to something like medical school, and some people go to graduate schools. Basically, what that is I said I really, really liked studying physics, and I want to keep doing it, and I want to learn even more physics. So now I'm doing even more school, and instead of kind of learning about science, a lot of what graduate school is learning how to be a scientist. And so now I'm a graduate student at Yale, and as I said, I study physics. Now, I've said that I study physics several times now, but I also, in my description of the talk, said we're going to talk about neuroscience. And so those might not seem like they go together. So does anyone want to kind of tell me what they think of when they think of physics? You can just type some stuff in the chat. I'll just read off maybe the first couple that go in. It can be absolutely anything. Just what's the first thing that comes to mind? All right, nukes, forces, gravity. All right, yeah. So falling, yeah, a lot of these things all have to do with physics, but motion is another good one. And so when we think of physics, I like to think of it as the way things move and how things interact with each other. So that does have to do with forces. It has to do with how things touch. It also has to do with energy. So people are talking about things like nuclear power, that all of that energy is very related to physics as well. Now, what do you think people think of when they think of biology? Life. Yeah. Earth, life. Yeah. So a lot of times when we say biology, we think of life, the study of life, living things. And so like plants, animals, pets, yep, or lots of biology going on in our pets. So all of that is going on. And then finally, what are a couple things people think of when they think of as chemistry? Explosions, chemicals. All right, you guys, solutions, seeing more explosions going on, chemicals. We do have to be careful with chemicals, make sure we're not having any reactions we're not intending to. But yeah, we think of a lot of liquids, changing colors, test tubes. I feel like that's a lot of times the images we think of when we think of chemistry. But chemistry is really has to do, it is a lot to do with mixing, but we think of it almost at this level of like, what do different types of atoms and molecules, how do they interact with each other? And so I think especially in like K through 12, we view these three big science disciplines as kind of very separate. And I personally, and that would put me over here in physics, but neuroscience over here in biology. And so that means how would I be, as a physicist, be able to look at neuroscience? But I think in reality, I think the sciences look a lot more like this. I think there's a lot of overlap, which I think is really exciting. And so when we look at neuroscience, yes, it's the study of the brain, it's the study of the nervous system. But the way the nervous system sends information throughout the body is using electrical signals. And so that has to do with a lot to do with physics. And there are lots of forces. Our cells are physical things. So there's going to be lots of different forces, lots of different movement going on in our bodies. So that's another way we can think about physics too. And also, truly, I think we could even argue that if I'm studying neuroscience, we could say that I might even end up here because there's lots of different chemicals that are being used in the brain. So I just like to put this out here as this is how we get to, I think, really cool projects that I get to work on where even though I study physics, I get to do biology. And yeah, I see some really great comments going on about how a lot of these things are really similar and we get to learn about them separately, but I think it's also important to see how they work together. And so I want to talk now about what I like to study. So I said this talk is going to be on visual processing. And so what that means is the way we see is that light enters our eyes. It hits the back of our eye called the retina, and then the retina sends signals through the optic nerve to the brain. And so it goes through all of our brain cells. And if you have never seen what a brain cell looks like, this is kind of a cartoon. They can look kind of like this. This is a pretty standard model picture of what a brain cell called a neuron might look like. And then once it goes through all of these cells, it gets to a certain part of our brain that then we can see either an image, we can have an idea. In animals, right, if a predator is coming at an animal, that's an image they're going to see, and it needs to get through their brain very quickly to tell them that they need to run away or hide from the animal. Yeah, and so someone said into the visual cortex, and that's right. And so we can talk about like different parts of the brain, but we're going to actually focus a little bit more not on the parts of the brain, but actually on some of the cells that work to do different tasks. And in this case, we will be talking a lot about the we will be talking about things going on in like the visual cortex, and in fruit flies we call it the optic lobe. All right, so how do we learn about things in the brain? So I mentioned that I work with fruit flies, so I like to study fruit flies. And does anyone guess why we might want to study fruit flies to learn about the brain instead of maybe a human? Someone said that they have good eyes. They definitely have different eyes. Any other guesses? They're very similar, easier to dissect. That's actually a very interesting idea of thinking about how easy it is to dissect a fly brain. I will actually say fly brains are very, very small, at least compared to a human brain. But what actually is the most interesting thing is not just that flies are maybe smaller and easier to do experiments on. They actually have they have a less complex brain than humans. So let's see, let's get a couple guesses. How many neurons, so brain cells, do you think are in the human brain? 110, a lot. Hundreds of thousands, trillions, more than 10, definitely more than 10. All right, so it is about 86 billion neurons that we have in the human brain. All right. And so how many neurons do you think we have in the fly brain? Oh, yes, so it is definitely less. All right, we've got lots of good guesses, got thousands, tens of thousands. Okay. So fly brain is about 100,000 neurons. So definitely still a pretty that's a lot to deal with, but it's a lot less than in humans. And so we can kind of work with a smaller system, learn how some of these things interact, and then look for patterns that might still apply even in a bigger system. And so how some of the things we might do with this. So I work in Damon Clark's lab, and this is how we are able to do this. So basically we have our fly in what I like to call like a VR, like movie theater. So we have it standing on a ball and it can walk on the ball, but it's being held in place. So it's almost like a treadmill. And then the screen around it is then kind of projecting movies onto it. So it's gonna, we got the fly, we're looking at a screen in front of it, and then we can show it movies. We can show things moving in front of it. Then we can see how the fly moves in response to it. And so we can either see how it moves, or we can, as someone was saying in the chat, we can take pictures of its brain. So we'll get to that later. But I also wanted to show you actually what it looks like. So this is kind of taken from behind. So it's being held in place on its little treadmill. And then you can see it's out of focus in the back, but you can see that there's actually moving bars on the screen in front of the fly. And then we are able to measure how the fly is moving by seeing how much that ball moves as they walk on it. So that's one way that we can look at actually how they move in response to different movies. But we can also then look inside the brain. And so we got a picture of a fly brain, but let's actually zoom in. So we're just looking at one cell. And so as you saw, there's a microscope above them. And so what we're able to do is we put special chemicals, sorry, we were talking about how chemistry is involved in here too. So special chemicals, we have them that they're attached to a certain cell. And then what happens is once it's, let's see, we have it. We have this special chemical, when we shine a laser on it, it actually glows. And so we can see that glowing under the microscope. And so basically when a neuron is being active, it glows brighter. And that tells us that it's being used. And so that way we can tell that certain cells are being used by how much they're glowing when we look at them under the microscope. And let's see. Oh no. Give me 1 second, guys. That, you know what, actually it's see-through, so this is gonna work anyway. So we have a single neuron here. So the way it works is it has inputs and it has outputs. So basically the kind of circular side of the cell takes information in, and then it does something to it, big question mark, and then sends output down to the next cells in order. And so in real life, the signal is an electrical signal, but we can kind of, simplify this down into, let's say maybe the cell knows that if it gets a heart signal, it's going to output a triangle. If it gets a star signal, it should output a circle. And so in this case, say the heart signal goes in, then we want a green triangle signal out. But this is very simplified down. Obviously, we're not dealing with just two shapes and two outsides. What this is really is just kind of a big black box. Which is what this question mark is that we don't know what actually is happening, but we do know what inputs we're showing the flies. We know what movies we're showing them. And we know what they're doing. So we can see how they're moving on the ball, or we can see how much the neuron is lighting up under the microscope. And so then we can try to work backwards and figure out what's going on here. So what math should be going on here? Is it a red heart equals a green triangle, or maybe not? But the thing is, this is, again, a much simplified version. In real life, it's a big network of not only is there lots of inputs and outputs, they're talking to multiple cells. And one of the other cool ways that we can learn more about this is we can take the cells and we can say, I want to see what happens if we take one cell out of the network. So now this cell doesn't work anymore. And now we can see, well, does the fly still walk on the ball the same way it did before? And that is one way we can actually study, because if it moves differently without this cell in place, we know that that cell must be pretty important to understanding understanding that part of the network and how it sees and responds to that one thing. And we can do that with multiple different cells in the network. So again, we could say, all right, well, what about this cell? How does this one change? How the fly responds to different movies in our little movie theater setup. And so these are a bunch of different ways that we do these experiments, because again, we want to know how they take different images and make them into something useful in their head. And so, like I said, we can show them different types of movies and then see how they respond, or we can show them the same movies but change the way their brain works a little bit. And now let's talk about other animals. So I said I would talk a little bit at least about mice and flies. So let's start really simple. What are some differences between mice and flies? No wings. Vertebrates. Love that answer. That's right. Let's see what else. Mice are furry. Yeah. So not only are mice vertebrates, they're also mammals. Yep. And flies are insects. Yeah, they have different anatomy, different types of mouths, they're different brains. And we talked about, like, even their eyes look different, right? Oh, and that's a really good question someone brought up. Mice do have a more complex brain than a fly. I believe they have somewhere in the 10 million-ish number of neurons, although I can't remember off the top of my head, approximately. So it's more than flies, but less than humans. And but yeah, so we have all of these differences in flies and in mice. But what's interesting is these are two pictures of some different, one side is a mice cell and one side is a picture of fly cells. So let's see, let's have a guess. Do you think the fly cell is on the right side or the left? All right. I honestly, I can't keep track. I feel like it seems pretty split. But so I will tell you that the picture on the left side, this is actually a picture of a mouse cell. And this on this side is the picture of a some fly cells. And so specifically right here where it says T4, this line right here, and T5, these are the cells we're most interested in. And what's interesting is these two cells, T4 and T5, they are very similar in terms of how they take input and output as this mouse cell. So if we think back to our, our really simple version where we're taking what shape input gets us the same output. Well, in mice, we can take the input here and get a similar output as if we did the same thing with these two cells in flies. And the numbers in the bottom are the size of these pictures. I can talk about that more later because we are running tight on time. So the last thing is that I did want to mention is how we do things in humans. So obviously we can't look at individual cells in human brains quite the same way, but we can ask humans what they see when they look at either pictures or images. And so this right here is we have two different little videos. And if you look at them, it kind of looks like they are, the static has movement. And so the top one kind of looks like the dots are moving to the right, and the bottom one, it kind of looks like the dots are moving to the left. But what's interesting is that these are actually about the same, these are the same video, except we switched which dots were white and which dots were black. So this is an illusion that we have been able to see in a lot of different animals, including flies, including mice, including humans, that even though this static is created in exactly the same way by just flipping which ones are black, which dots are black and which dots are right, they look like they're moving in different ways. And I do have one other really cool version that uses this, but it flashes, it's a little bit more flashy, so I just want to put a quick warning before I show it because I don't want anyone to be caught off guard. But I can show you, let's see, very quick, because I think it's really cool. And this is the last little frame that I have. It's actually, it's a little video created from 4 frames. Nope, not this one. I'll come back to that one later. So I'm gonna stop my share real quick because it's not, I can't see what I'm showing you. Let's see. Sorry for the delay, and hopefully this works now. All right, yeah. And so this is our last little thing. So what's actually happening is there's only 4 different frames. There is a white frame and a black frame in 2 different positions. And so just by switching between those 4, it looks like they're moving, and then the arrows are kind of suggesting what way they're moving to you. But it's really the cubes themselves only have 4 different, or really 2 different positions, and then they're just switching if it's black or white. And so that's another example of how we can use this specific illusion. So I'm going to stop sharing. And thank you guys. That was actually the last illusion I wanted to show you guys before we go into our breakout rooms and actually talk a little bit more about some illusions. But I hope this was interesting. I know I talked about fruit flies probably a lot more than you were expecting, but fruit flies are really, really cool scientific tools.
Study neuroscience is important?
Oh, that's a really great question. I think that there are so many wow, I have a lot of thoughts on this. So what's interesting is because for so long, those kind of 3 major disciplines of science have kind of been taught separately and studied separately. There's a lot of like ideas that didn't necessarily like get communicated between those different disciplines. And I think so part of it is that just getting the different ideas that might be really, common in physics and taking them over to neuroscience can really help answer some of these questions. And it's kind of like just a diversity of perspective. But I also think that when we have different experts coming together, we have just more overall knowledge, which is really useful. And like I said, there's lots of aspects of neuroscience, like the way electrical signals travel, that are really rooted in physics, but otherwise might not grab a physicist's attention otherwise. So I think it's really important that we have all the different groups come together, like in the Venn diagram I showed.
Yeah, I think that's a very fascinating answer. I think that really highlights the importance of having diversity of perspective and also collaboration in science in terms of, putting successful science into the world, right? So, the second question, I guess, is a three-part question. So, why do you study eyes, and why do you want to study that specific question, and what inspires you to do that?
Oh, well, I think that um, well, so vision is a very important sense to a lot of animals. So there's obviously animals that either don't use their sight as much, but for animals that do have sight as one of their primary senses, it's absolutely like essential to survival. And what's really interesting is kind of like what I was starting to say a little bit with mice and flies, is that we can see patterns and similarities across a lot of different animals that you might not see. Like we said, like we were saying, like mice and flies are very different animals, and even the cell shapes look very different. However, if we go down to this question I was asking of how do we actually process um, light into useful images, that's where we can see these similarities. And so that I think is something that will answer not just this, question of how do we process visual things with our eyes, but how do the neurons themselves, how have they like evolved to solve this problem of being able to see? And I just think that's really cool. I think that was the last part, is why do I, or one of the parts was like, why am I interested in it? And I just think that's really cool that, I don't know, like, yeah.
Yeah, I mean that's an awesome answer, I think.
Yeah.
Yeah, I think this stuff is really amazing. So then the next question is related to technology. So do you think that retina display technology is related to how the retina works, or like inspired by the retina?
So this is going to be kind of embarrassing. I don't actually know how retina, like what that actually means in terms of technology. So I can't actually answer. Um, but I would imagine that the scientists that came up with it probably had some sort of inspiration, but that's also another example of why collaboration is important, right? Is that we um, maybe they whatever ideas they got, they might have gotten from biology and how a retina works.
Yeah, yeah, yeah.
So,
Yeah. And the next question is related to your choice of using fruit flies. So
mm.
um one really really good question is like why do like we have to dissect frogs so we can just study fruit flies, like.
Okay, well, so in biology, we like not just for neuroscience, but lots of different biological questions. There are a lot of different, we call them model organisms. So basically, we can't always be doing research on humans or on even on primates or even on mammals, but different types of research questions might be served better by different organisms. So for the type of vision research that I do, there's a lot of really interesting things we can do with flies. Like I was saying, we can kind of uh, like have the brain ignore certain neurons and then still see what responses happen. So that's something that we can't do in a lot of other animals. So that's something useful. Um, there's a lot of really interesting like genetic tools besides that we can use with flies that I don't have time to explain here. Like I said, for the type of kind of small system network mapping that we're doing, flies are a good size brain, but there's lots of other biological questions that use different animals. Like I know that thank you, moderator Lom works in a lab that actually works with bacteria. And then I also have friends who work with primates and monkeys because their research questions are better suited for those types of animals. So it just depends on what you want to answer.
Yeah, that's awesome. And another question is, can you dissect fruit flies? Like, do you like how do you keep them still, and what do you do with them after?
So I wouldn't say that we dissect them, but and I should also say that I personally don't do fruit fly surgeries, but some of my talented lab mates do. So for keeping them still, we're able to knock them unconscious basically using CO2. So the same, kind of similar if you go to the dentist and you get laughing gas. While we give them CO2, which makes them sleep. And then we have special little tiny, I guess, I don't know, it's a little holder. And we can take the fly and put it in the holder while it's asleep, and it holds them in place. And then they are able to actually put them under a microscope, and they use the little tiny needles actually. And they can do little surgeries, and that's part of how we're able to take those really cool pictures of their brain, is that we just do a little tiny surgery and actually shave off part of the exoskeleton on the top of their brain, or on the top of their head. So then the microscope can see right into the brain cells where, like I said, we have the chemicals that glow under the laser.
Wow, that's super cool. I didn't know.
It's really cool.
I do know that they do fly surgery, but it's such a cool concept. Doing
Yeah,
that, imagine doing surgery on that tiny little creature, right? It's amazing.
yeah.
And I guess like the last question is, so in the network of like neurons, and you said that, scientists is able to, pick out what neuron could be responsible. So how did they able to do that in the flies?
All right. So yeah, so this is getting into some of the genetics stuff. If you've heard of CRISPR, it's been kind of big and talked about more in like pop science, but basically we're able to use these kind of special molecules to like modify the DNA a little bit of these flies, and so that they actually um, either don't have that specific neuron, or that specific neuron is there but it doesn't work, or doesn't work in certain temperatures. So that way, if we then have the flies living at a certain temperature, that neuron isn't able to work in that temperature. And that is all done by not by us. There are labs that do that, and then we are able to actually order fruit flies from them that have those specific genetics. And so then we have the flies we need. But they do it by kind of trial and error, of going through trying these different genetic editing and then testing it to see. So it takes a really long time, but it's really cool what they're able to do with it. And that's why fruit flies are very good for some of these experiments.
Yeah, that's awesome. That makes me want to work in fruit flies, and I hope you guys here will join grad school to study fruit flies genetics and also vision. So thank you so much, Caitlin, for providing an amazing talk. We really enjoyed it, and I guess I'll transfer it to the actual volunteer to end our session.
Yeah, thanks everyone for coming. Let me just quickly share my screen here. All right, so thanks everyone for coming, and thank you so much to Caitlin for that really cool talk. I know my breakout room had so much fun with the optical illusions. And for anyone, I know a lot of people in my breakout room were actually new to exploring science this semester. So if you want to catch up on any of the previous talks that you didn't attend, we have those all online. So you should go check those out because those were all really cool as well. And then next month, our next Exploring Science talk will be by my friend Yara, and she's going to give a really cool talk. It's interesting that people were asking questions about frogs today because she's going to be talking about how frogs are able to survive freezing during the winter. So like certain types of frogs can actually completely freeze solid during the winter and then thaw in the spring and live totally fine after that. So how are they able to do that? that will be the topic of next month's presentation. So I hope you can all make it there, and thanks for coming.